Light microscope. Microscopy is a technology of making very small things visible to the human eye. The instrument used for this technique is called a microscope, in which we can use visible light to make specimen observable. The discovery and early study of cells progressed with the invention of the microscope in 1590 and its refinement during the 1600s. In 1665, Robert Hooke observed the cell wall of dead cells from the bark of oak tree through a microscope. The different types of microscopes are the optical microscope, electron microscope, and scanning probe microscope. Optical microscopes function on the basis of optical theory of the lens by which it can magnify the image generated by the passage of a wave through the sample or reflected by the sample. In the electron microscope, the electron beams are used for magnification. The basic optical microscope system used in the laboratory is a light microscope in which visible light passes directly through the lenses and specimen. Light microscopes can be categorized into the bright field microscope, phase contrast microscope, dark field microscope, and fluorescence microscope. Three important parameters in light microscopy are magnification, resolution, and contrast. The microscope consists mainly of three parts. The mechanical part of the microscope consists of a base, C-shaped arm, and a stage. The magnifying part consists of an objective lens and an ocular lens, and the illuminating part consists of substage condenser, iris diaphragm, and light source. Materials required: light microscope, immersion oil, prepared glass light with sample. Procedure: first, switch on the light source and adjust the light intensity with the help of a rheostat or brightness control. Place the prepared glass light with the sample on the stage of the microscope and rotate the nosepiece to the lowest power objective, that is at 4x objective lens. Observe the sample through the microscope and adjust the iris diaphragm to the largest diameter to make the background brighter. Then close 3/4 of the iris diaphragm and adjust the coarse adjustment knob until the specimen is in focus. Now rotate the fine adjustment knob to view the image more clearly with good resolution. Again rotate the nosepiece to the 10x and 40x objectives and repeat the same steps to focus the specimen and to get a finer and clearer image of the sample. Then rotate the nosepiece of the microscope in such a way that the specimen is in between 40x and 100x objectives and add a small drops of oil on the center of the slide. Again, rotate the nosepiece such that 100x oil immersion objective touches the oil. Then open the iris diaphragm completely and adjust the fine adjustment knob until the images become clearer. Remove the slide and wipe the 100x oil immersion objective carefully with tissue paper to remove the oil. Finally, switch off the light source. Applications: the light microscope is used extensively in cell biology, microbiology, biotechnology, pharmacology, and forensics. It is also used for medical diagnosis, particularly while dealing with tissues or in smear test on free cells or tissue fragments.